Good morning, students. This is the last session of the human reproductive system, in which we study about the fertilization, implantation and embryonic development. You can see this video on my Facebook page Word of Biology 95.wordpress.com, and you can subscribe this channel also. Now, fertilization takes place after the 14 to 24 hours of the ovulation, or 12 to 24 hours of the ovulation. Actually, the male gamete or sperm can remain viable about 48 hours after the ejaculation, and the female gamete or secondary oocyte is viable for 24 hours after the ovulation. But the chance of fertilization can be extended up to 72 hours of the sexual intercourse when the ovulation is taking place or ovulation has taken place. The sperm produces an enzyme that is called as acrosome. Actually, we have studied earlier the sperm has acrosome that has an enzyme like the lysozyme, that is also called as acrosome, and this acrosome is essential for the fertilization. This enzyme stimulates the motility and migration of sperm also. Just after the ejaculation, sperms undergo capacitation in the female reproductive tract. Female reproductive tract means from the vaginal canal till the fallopian tube, because the fertilization is taking place inside the fallopian tube at the ampulla-ischemic junction. So the male gamete or sperm starts its capacitation during the female reproductive tract from the vaginal canal to the fallopian tube, and during this capacitation a membrane around the acrosome gets fragile so that the several destructive enzymes are released. During this capacitation acrosome का जो membrane है वो destroy होता है, और after the destroying of the membrane जो एंजाइम्स होते हैं वो रिलीज आउट हो जाते हैं, और रिलीज होने के बाद these enzymes they penetrate the corona radiata as well as the zona pellucida layer of the oocyte. Corona radiata, that is the group of cells found around the ovum or female gamete, and zona radiata is outermost layer of the egg cell or the ovum. So by the help of acrosomal enzymes or acrosomic enzymes, fertilization is getting possible. Here the female gamete is surrounded by numerous male gametes or sperms, from which a single male gamete becomes able to fuse with the female gamete. Penetration of a sperm stimulates secondary oocyte to complete meiosis II. Now, we have studied earlier the female gamete is released from the ovary at the secondary oocyte level just after the first meiotic division of the primary oocyte. During the fallopian tube, when the sperms approach the female gamete for the purpose of fertilization, it triggers the second meiotic division or maturation division of the secondary oocyte to produce female gamete and second polar body. Fusion of ovum and sperm is called as syngamy. Syngamy means the true fertilization when the male gamete and female gamete are really fuses together. Syngamy causes the depolarization, which triggers release of calcium ions into the cell, and the calcium ions stimulate changes in the zona pellucida layer, which prevents the polyspermy. What is the polyspermy? Actually, polyspermy means the fusion of male-female gamete along with the more than one sperm. But it is not taking place, it does not take place in the case of male reproduction. In the case of human reproduction, only one sperm is able to fuse with the female gamete. When the single sperm fuses with the female gamete, then certain changes are taking place, and after these changes, the vitelline membrane of the egg cell or female gamete gets changed into fertilization membrane and checks the entry of other sperms to the fertilized egg. So, the zona pellucida layer checks the entry of the sperms. The vitelline membrane is getting converted into fertilization membrane that checks the entry of a sperm. So, finally, single sperm is able to enter inside the female gamete, not more than one sperm is able. So, the polyspermy is not found in the human reproduction, it is the process of monospermy only. Now, after the fertilization, ovum is considered a zygote, and here the post fertilization changes is shown in this diagram. That is the zygote. Inside the zygote, cleavage starts after the fertilization. 2-cell stage, 4-cell stage, 8-cell stage, morula, that is the solid football like structure, and after the morula the blastula stage is developed, or blastocyst stage is developed, in which the cells of the zygote or embryo, cells of the embryo are arranged at the peripheral positions, and rest of the cells are accumulated at a particular point, and a larger cavity is formed inside the blastula. This larger cavity is called as blastocele or blastocyst cavity, and these peripheral layer or these peripheral cells are called as trophoblast, and this accumulation is called as embryoblast or inner cell mass. So, this is the blastula stage, morula stage. These are the different changes taking place after the fertilization. Now, after that, at the stage of blastula, the embryo is getting implanted in the endometrial wall of the uterus, and this process in which the embryo is attached with the endometrial wall of uterus is called as implantation, and this implantation is taking place usually on 7th day after the fertilization. At the 7th day of the fertilization, how the implantation is being completed? Now, see this diagram. Here is the development of placenta, and this is the uterine structure. This is the endometrial wall, and along with the endometrial wall, this structure is getting attached. At the 7th day, the trophoblast after the fertilization, the embryo is getting developed, and during the development of embryo is started moving towards the uterus, and on the 7th day after the fertilization, embryo is getting attached with the uterine endometrial wall by the help of placenta. Actually, trophoblast performs the important role in the process of implantation. The trophoblast cells gets embedded in the endometrial wall, and finally the implantation is completed. After the implantation, a female is said to be pregnant. Before the implantation, female is not said to be pregnant. It means fertilization is not the confirmation of pregnancy. After the implantation, pregnancy is confirmed. Here is the structure of placenta, and in this placenta we can see the different fetal membranes also. That is the chorion, amnion, allantois and yolk sac. These are the 4 fetal membranes or embryonic membranes found in the human fetus or human embryo. Amnion, that contains amniotic fluid. Chorion, that has so many finger like progressions. These are called as chorionic villi. These chorionic villi are getting embedded in the endometrial wall of the uterus. And this disc like complete structure is called as placenta. Inside that, the human embryo is being developed. Now, during the pregnancy, the embryo receives all the nutrients, oxygen and other useful materials from mother through the blood, and all the waste materials from the embryo and or fetus are released out through the blood to the mother body, and this connection is done through the umbilical cord. Actually, umbilical cord is the connection between the mother and the fetus. So this is the structure of placenta. Now here you can see that this diagram. Here this is the umbilical cord, and these are the umbilical vessels. This is the complete contact of the placenta with the endometrial wall, and inside the placenta these are the chorionic villi, and inside the chorionic villi these blood capillaries are spread. These are the maternal blood vessels. From maternal blood vessels, all the materials are exchanged through the umbilical cord to the body of the fetus. So it is the structure of placenta. Gastrulation, actually after the blastulation, just after the implantation, the next stage of the embryonic development, that is called as gastrulation, and this gastrulation is taking place. This gastrulation means the development of the different germ layers. Since we are the triploblastic in nature, so after the blastulation, when the gastrulation is taking place, the 3 germ layers are developed, and these 3 germ layers are ectoderm, mesoderm and endoderm. These 3 germ layers are developed by the arrangement of the trophoblastic or inner cell mass of the blastocyst. Now here the 3 germ layers, ectoderm, mesoderm and endoderm. From the ectoderm, nervous system and epidermal system of skin are developed. From the mesoderm, skeleton, muscles, circular system and gonads are developed. From the endoderm, digestive tract, respiratory tract, liver, pancreas and bladder are developed. These are the major organs which are found inside our body, and these are developed from these 3 germ layers, ectoderm, mesoderm and endoderm. So this is all about the development of the organs from the 3 germ layers after the gastrulation. It means we can say that after the fertilization of zygote is converted into morula after subsequent cleavage. Morula is converted into blastula or blastocyst. Blastula is converted into gastrula, and after the gastrulation, the organogenesis is being started. The organogenesis is the result of the gastrulation, when the embryo is developed into the fetus and their fetal organ or their body organs. Now, how embryo develops into the baby? The first trimester, the brain begins to form in 2 weeks old embryo. Between 3 to 4th week, heart begins to beat. Between 4 to 6 week, vertebrae and ribs begin to form. By the 7th week, nervous system begins to release signal. And in the second trimester, the skeleton made of cartilage changes bones. Any cartilages are converted into the bones. External genitals begin to form. Hair starts sprouting out. In the third trimester, at the beginning of the third trimester, fetus open her eyes for the first time. Rest of the time is dedicated to storing fat, gaining weight and practicing breathing. So this is the all about fertilization, implantation and embryonic development. Thank you.